Hello students, so now we are going to discuss about the sources of drugs, right? So when we talk about the sources of drugs, basically it may be from the plant source, it may be from the animal source, it may be from the marine source, and maybe from the tissue culture, right? So these are the four major sources which is there into the syllabus also, right? So one by one we will discuss here, right? So when we talk about the plant sources, right? So what do you mean by plant source? Of course, the name itself is indicating that the drug obtained from the plant origin or plant kingdom, right? So it is the oldest source of the drugs. Even in history of pharmacognosy, we have seen that right from the beginning, the people, the human being, they started using the plant kingdom for their food source of, then they later on for the medicine, right? Most of the crude drugs derived from plants in ancient time, right? So the drug which was mentioned into their Charak Shamhita, Sushruta Shamhita, and Papyrus Ebers, in so many history parts we have seen the scripture available in ancient time. There is a there is a description of the plant origin, so much of the crude drugs, right? Almost all part of the plants are used to cure, right? It may be root, stem, fruit, flower, bark, maybe entire plant. It is useful for the cure of disease. So there are examples like leaves we are using, roots, bark, fruit, stem, etc., right? So we are going to discuss about the different examples so that it will be impressive answer, right? So coming to the first one, when we talk about the leaves, right? So what are the leaves parts we are using? The example is very important when you are describing this kind of answer. Digitalis purpurea, right? So this is the one of the leaf drug where from their foxglove leaf also it is known as, we are getting digitoxin and digoxin, these two types of cardiac glycoside we are getting, right? Then we are having eucalyptus, which is useful into the cough and cold syrup preparation. Then from tobacco we are getting nicotine. These things we have seen into the history part also. It is useful as a natural pesticides and antimicrobial property. It can kill the microorganisms, bacteria, fungus, etc., right? Atropa belladonna, right? Belladonna leaf, it is very famous. Atropa belladonna from here, from this leaf we are getting atropine, right? So this atropine, it is useful in the dilation of eye pupils and so many function, pharmacological action into the eye, right? Coming to the root part, when we are having a root example, Rauwolfia serpentina is the root. Rauwolfia root is very famous. From there we are getting reserpine, which is useful into hypotensive agent. Then Withenia somnifera, withenolites, right? Then coming to the bark, we are having so many other example like cinchona bark, cinnamon bark. In cinchona bark we are getting quinine and quinidine, which is useful into the anti-malarial. Then cinnamon bark, in the cinnamon bark we are getting volatile oils, and that is useful into the carminative. Then coming to the fruits, in fruit part we are having so much of the fruit, for example, we are having this one coriander fruit, right? So when we talk about the coriander fruit, we are having this thing, coriandrol, and volatile oils can be obtained from coriander fruit, that is useful into the carminative. Then fennel fruit, fennel sopu or sop, that we are saying, right? Volatile oil containing crude drugs, carminative effect. Flowers we are having vinca flower, vincristine and vinblastine is the very famous anti-cancer agent. Then pyrethrum flower we are having pyrethrin, we are getting pyrethrin 1 and jasmolene, that is useful as a insecticides. Then stem parts also we are using in ephedra. Ephedrine is the constituents which is useful as a bronchodilators. Then in wood parts we are having kochia wood. Kochia wood is very famous for the quashin, right? Quashin, that is having anthelmintic activity, and it is also useful as a bitters. Then seeds, nux vomica, strychnine, brucine, you can write down, and it is natural pesticide. So these are the series of the parts of plants which we are using as a crude drugs, that you can mention in your answer as a example, for example, right? Coming to the second source, as per this thing, we are having animal source, right? So when we talk about the animal source of crude drugs, we can have this one, right? So just a minute. Right, animal source. So when we talk about the animal source, these are the primary and crude type of drugs which we are getting. These are the primary and crude type of drugs, such as hormones we are using, oils we are using, biological fluids can be useful, obtained from the animal body. It may be ox, sheep, pig, or so many other animals. We can use cattle and all. Examples, we are having pancreas, right? So any of the ox pancreas can be useful for the isolation of insulin which will be useful into diabetes. Those who are suffering from diabetes, they can be given this particular insulin. Thyroid, from thyroid gland we can isolate from the particular animal, and thyroxine hormones can be isolated, and it can be useful into the hypothyroidism, right? Then hypertension also. This cardiac cod liver oil, we are using oil which can be given to the vitamin A and D deficiency people, and that particular thing can be complete, right? Then coming to the other examples, we are having animal bloods also can be useful into the preparation of so much of vaccines, right? Then pituitary gland, from there we can get the gonadotropins, can be useful into the infertility case, right? Then third source, the major source, that is the marine drug, right? Marine organism, marine foods are very famous, right? So when we talk about the marine organism, these are the substances of medicinal value obtained from marines. It may be sea or ocean sources, right? So in like we are having plant and animal source of crude drugs, like that inside the sea and ocean there are again plant and animals and organisms, microorganisms, so many things. But because sea, the marine world is totally different, right? They are also having plant, animal, microorganism, and mineral, right? So from those things, how we can get the different types of moiety which can be useful into the therapeutic property purpose. Examples, we are having first one, marine sponge, right? So from marine sponge we are getting ERA A, which can be useful as a antiviral. So this is the series of the example you are supposed to write when you are attempting this kind of question. And then example B, sponge and gorgonians can be useful, right, to isolate the euristomines, right? So it is antiviral again. Then third example, marine fungus can be useful for the isolation of cephalosporin C, right? That is nothing but antibiotics. Then red algae can be useful for to get the laminin, which is anti-parasitic drug. Then marine organisms can be useful for the bryostatin isolation, which is anti-cancer. Then octopus, all of you know the starfish, octopus, these are the main part of the sea and ocean. So from octopus we are getting octopamine, that is nothing but cardiotonic property it is having, right? Then marine microbes also can be useful to get the butanolides, and which is nothing but anti-inflammatory drug, right? So like that, the fourth one, the fourth category of crude drugs source, that is the nothing but tissue culture, right? So we are having tissue culture once again, right? It is in vitro cultivation of plant cells or plant tissue under aseptic and controlled condition. We are using this in suitable nutrient medium for the regenerate plant or for the production of primary and secondary metabolites. Ultimately our goal is to get the medicinal constituents or chemical constituents which can be useful into the therapeutic purpose, right? So that's what in laboratory. In vitro means in laboratory, whenever we are trying to cultivate by providing the suitable condition, control condition, aseptic area, and a proper nutrient medium, we are generating the plant. So what are the things we are doing? In example, you can write down atropine, which is obtained from Atropa belladonna. So Atropa belladonna cultivation can be done into the laboratory to get the atropine, which is by using the hairy root culture, right? Then the another example we can write here is asmalicine, which is obtained from Catranthus roseus, that we are doing by the suspension culture. Then third example, papan, carica papaya, we are using callus culture, right? So these are the three different types of culture can be useful. Codeine also can be obtained from Papaver somniferum and by the using the suspension culture, right? So these are the four sources of crude drugs which we are supposed to write in the exam with this proper example, right? So our this is what the kind of question will come. What are the different sources of crude drugs? Explain them with examples, right? So here whenever you are describing plant, animal, marine and tissue culture, you need to give the proper examples, then only your answer will be fulfilled. Thank you.